Going to start our amphibian experiments. There will be two types of amphibian experiment. One will be on nerve muscle physiology. Another will be on amphibian heart. We will be studying some properties of nerve and muscles through these experiments, amphibian experiments, nerve muscle experiments. For example, we know muscles are excitable tissues. When appropriate stimulus is given, adequate stimulus is given, adequate stimulus means threshold stimulus is given, then they will respond. This is called excitability. So this excitability of the cells are because of what? They are having a transmembrane potential. What is this called? Resting membrane potential. So these cells are having resting membrane potential. So when we give an appropriate stimulus to our nerve is also excitable tissues. So they get stimulated, they give response. How they give response? By producing action potential. This action potential is nothing but nerve impulse. And the nature of this nerve impulse is they are propagatory in nature. They will transmit along the membrane, along the neurolemma. Okay? So this is the response given by the nerves when they are stimulated. So we will also be studying, if I give a stimulus to a nerve, so it will produce action potential and this will be transmitted. Okay. You can determine what is the velocity of impulse transmission. Per unit of time they will be traveling to certain distance. What is this? Suppose if it is the sciatic nerve. What is the sciatic nerve? It is the myelinated nerve. We have classified nerves in various ways. According to either it is afferent or it is efferent. If it carries impulse from the periphery to the center, then we call it afferent nerves or sensory nerves. If they carry impulses from center to the periphery, we call them motor nerves. So this type of classification is there. Also, depending on the neurotransmitter secreted from their nerve endings, from the axon terminals, we classify them into adrenergic nerve, cholinergic nerve, and so on. Okay. We also classify them according to the presence or absence of myelination. So they can be myelinated, they can be non-myelinated. And most important that you may expect a question in drum.and or in assessment, that is nerve fiber types according to the diameter and velocity of nerve impulse. You will find that depending on the diameter, depending on the myelination, the velocity of nerve impulse through this nerve varies. Higher the diameter, higher will be the velocity. So you can determine the velocity of nerve impulse, particularly in amphibian experiment. You will do it. Another important thing is when these nerves are having one termination, it may be gland, it may be muscles, it may be cardiac muscle, it may be skeletal muscle, it may be smooth muscle. So this impulse, when this will be going to their target organ, there it will produce certain response. If this impulse is transmitted to the skeletal muscle, then what will be the response? Their contraction. This contraction may be of two types. One is What is this? Isometry and isotonic. So either it will produce isotonic or isometric contraction. So while we will be performing experiment with the amphibian muscle, the muscle we will be using is the gastrocnemius muscles. What is the muscle? Gastrocnemius muscle. And these gastrocnemius muscles are supplied with a nerve, this sciatic nerve. So they are originated from which segment? Lumbar segment. So from this, these nerves, sciatic nerves are originated and they are terminating into the gastrocnemius muscle. So to study the property of muscles, one is conductivity, another is contractility. Nerves are not having this property, contractility, because they are not having contractile elements. But the muscles are having contractile proteins, actin and myosin. So because of the presence of this contractile protein, they can show contraction. It may be, as I said, it may be isotonic, it may be isometric, whatever it is. So there, when this nerve impulse will be going to the muscles, they will show contraction. So contraction is another property of the muscles, contractility. We will experimentally see. Let's see, put some stimulation on the nerve, whether it can produce contraction in the muscles. So this you have to show it. You learned it in theory, now you have to show it through some experiments. So here is the ideal experiment is with amphibian muscles. So before we do it, this experiment with amphibia, we study the properties of the nerve and muscles, we have to prepare the nerve and muscles. And this is isolated. This nerve and muscles is isolated from the frog. And how it is isolated? How it is isolated? So this is the frog. You need a needle. You need a needle and a frog. So first, you have to stun the head of the frog on a hard surface. Then you can use a rod to stun the head so that it becomes unconscious. Okay. Then what you have to do? Use a needle, and you have to insert this needle through great foramen at the surface of at the neck of the frog. So then you can insert it into the vertebral column. Then you will feel, if you insert it, then you will feel hard, some hardness, and if you force it further, you will find some soft. Suddenly it became soft. That means you reached spinal cord. Within the vertebral column, what is the spinal cord? So you reach the spinal cord. Then what will be? This is the frog. It is horizontally kept. So you inserted the needle, then you incline the needle and push it through the vertebral column. And this will be, while going through, it will destroy the spinal cord. As a result, what will happen? All the ascending and descending nerves will be damaged. So there will not be any sensation, carrying of any sensation to the center. Okay? Then these muscles will be completely paralyzed. Okay? This center spinal cord will not be having any control over the muscles. Then again what to do? Now suppose this is the head. Then again you have to incline the needle this way. Then you push it towards the head and do the needle like this. So this will destroy the brain. So this process is called pithing. P-I-T-H-I-N-G. The process is called pithing. So after that, your function is to isolate the sciatic nerve and the muscles. So how you can isolate it? So this is the process. Since you will not be doing it, so I'm directly coming to this. This is the gastrocnemius muscle. This is the knee portion. Here is the part of, this is the part of the femur, and this muscle is attached with this bone through tendon. Okay, and this part, another part of tendon is there, which is attached to this ankle. So this part of this tendon Achilles. So you are having tendon Achilles and gastrocnemius muscle, and this is the sciatic nerve, and this is the part of the vertebral column from which the nerve comes out. So this part we call it nerve muscle preparation or gastrocnemius sciatic nerve preparation. Now this, you have to perform experiment with this. To perform the experiment, you need some time. So if you keep it open, in open air, you cannot keep it in open air or in water. If you keep it in open air, what will happen? The muscles, the nerves, they will be dried. So they may lose their activity. If you keep it in water, what will happen? Water means this is hypotonic in respect to the tissues. So you know what can happen? So again, the cell size will be changed. What will be there? Whether there will be swelling. What happens? This outside is hypotonic in relation to inside. There will be osmosis of water from outside into the cells, so they will swell. So the morphology will be changed, they may lose their activity. So we cannot keep them in water also to prevent dehydration. So what we have to do? So we can keep the solution in an isotonic solution. So for amphibia, the isotonic solution, there is an isotonic, so we can prepare, that is called ringer solution. What is this? Ringer solution. Now, we have to make this ringer solution. How can you make this ringer solution? This way you can make the ringer solution. These are the composition of, you need sodium ion, sodium chloride to maintain the osmotic pressure of the fluid, and you need also potassium chloride for maintaining excitability of the tissues, and for calcium chloride, these are needed for contraction of the muscles, and other two, these are buffer. Sodium bicarbonate and sodium hydrogen phosphate, these are the buffer. So they will maintain the pH. What pH? The cells are having, the tissues of the amphibian cells, they are also having a particular pH. This pH is similar. So there will not be any alteration of pH. So no alteration of osmotic pressure. So there will not be any change in their morphology. Okay. So maintain the external environment. With this, we are maintaining the external environment with this fluid. Is that clear up to you, right? Okay. Now, we need a setup. Now we need a setup, experimental setup. The first thing what we need prepared nerve muscle preparation, and how it is prepared, that we came to know. And next is the stimulation setup. How we will be stimulating the nerves. How will we stimulate? So for that, one setup is there, and the third setup is recording setup. The muscle will contract. How can we contract this? We can record this setup. Okay, and the other point is, so there will be a moving drum will be there. The moving drum, there it will be inscribed, the graph will be inscribed. So it is against time. Some contraction will be there against time. So you have to measure the time. How can you measure the time? You can measure the time on the speed of the kymograph, drum. The drum, movement of the drum is having a particular speed. That means it will travel a particular distance in a particular period of time. From that part you can also determine. But more accurately you can do it by using a tuning fork, which is having the tuning fork varies in their CPS. How many frequencies it can produce in 1 second. So here we will be using a tuning fork of 100 CPS. Means there will be 100 undulations, there will be 100 vibrations, 100 cycles in 1 second. So 1 cycle means how much time? 100 cycle means 1 second. 1 second means 60 divided by 100. Then how much it comes? This will be 0.05. So 1 cycle will get in the tracing. I will get a contraction, and this will be done through a period, over a period of time. So in this twitch contraction, there we will be getting 3 time periods. And how these all time periods you can calculate. Just you have to notice how many cycles are there in that particular time period. If there is 1 cycle, then 1 into 0.05. If there are 3 cycles, then 3 into 0.05 second. So in this way you can calculate the time period. Okay, so how many things are you require? One is nerve muscle, prepared mass muscle. Second is setup for stimulation. Another is setup for recording. And fourth one is setup for calculating time, estimating the time, measuring the time of different time periods. And that can be done by using a tuning fork. This is the recording device. The right sided one is the kymograph. This is the recording device. So this is the lower part. This is nothing but a motor. So this motor is having a shaft. This is the shaft. Within the shaft there is a drum. So within the outer surface of the drum, you can fix a white paper. You can fix a white paper. Now there is a method by which you can make it black. And the process is called smoking. How it can be done?
You can take the drum, you can take fire, like any candle or anything. It will produce smoke. So if you rotate the drum in front of the smoke, the first one can cover with smoke, and so it will be black.
Suppose this is the drum, that very drum. Suppose we'll have a piece of paper having this much width, and how much long? This much long. Now what we will do? We'll wrap this drum this way and put some gum here. That's it. Okay. Now what you have to do? You have to blacken it. Means you have to smoke it. How you can smoke? So there is another device. There is another rod-like device. You can insert it into the rod. And there is another handle. If you move this handle, it will be rotated. It will be rotating. Then you do one thing. There is a kerosene lamp. So fire it. So it will produce smoke. So with this kerosene, in one hand you just keep it on moving. Then with the kerosene lamp, you do it like this. So there will be, this is moving. So there will be a homogeneous layer of smoke on the surface. So now it is smoked. If you touch it, then at the background, the white background of the paper will be shown. So don't touch it. So then again you insert it on the kymograph. So this way you can make. This is for what? So for recording the twitch, for recording the muscle contraction. Next.
So this is the setup, the complete setup by we do the first experiment.
This is myograph. This is myograph. So the muscles we have to keep. Here we will keep the muscle. This is the myograph. This is the muscle. This is tendon Achilles. And it is not clear. This is having, it is having nerve, sciatic nerve. It is having the sciatic nerve. Okay. So now see, you pin it here. Before pinning, this is a lever. This is the lever. This lever is having a short arm and a long arm. At the junction of two arms, there is a pivot, fulcrum. Suppose this is the fulcrum, this is the fulcrum, this is the long arm, and this is the short arm. Okay, so now you see, if I pull it this way, what will happen to this? It will go up. This arm will go up. If I release it, this will come down. And if the drum is moving, this up and down can be recorded on a moving drum. I can record this up and down on the moving drum. So this is what? The stylus. At the stylus, this is a stylus attached with a lever. The short arm is attached, is having a thread. The other end of the thread is having a hook. This is, or without hook, only with hook, what you will be doing? You will be piercing the tendon Achilles. Okay, or you can tie it with a thread. Either you pierce it with a pin, or if you don't have pin or don't want to allow the pin, then you just tie it with the tendon and the thread. Okay. Then you do it, you pull it this way. In such a manner, pull it that much so that this arm becomes horizontal. If it is loosened, it will come down. If you pull it this way, then it will go up. So, in between, in such a place you pin it, the long arm remains at the center, horizontally. Okay? Now, there is electrodes. These are the electrodes coming from the stimulator. Coming from the stimulator, these are the electrodes. So, you place the knob over the electrodes. Okay, so this is the setup. And now you go for the stimulator. This is the stimulator. With the stimulator, we will apply some stimulus to the knob. And this stimulus is electrical. But we cannot give AC current, you cannot use the AC current, not the DC current directly, because they can damage the nerve or muscle. So what we have to do? So we need induced current. What is induced current? You have studied physics, you must know what is induced current. What is induced current? You forgot. Or you can remember.
Proper definition, it's like we induce the current using a...
You have to produce a magnetic field.
Yeah.
You have to produce a magnetic field. For how can you produce that? First you have to make a coil. With copper wire, you have to make a coil. And the two ends are connected to the power supply, DC current. Okay, so now if you switch on, then what will happen? This will be converted into, this primary coil will be converted into a magnet. And they will produce magnetic field. Now, if you place another coil, this is called secondary. This is the primary coil and it is the secondary coil. Then if you insert another coil over the primary coil, around the primary coil, then what will happen? This magnetic field will produce current on the secondary coil. And this current is called induced current. Then secondary current is produced in the secondary coil, one induced current is produced. We can use that induced current for the stimulation purpose. Advantage is it can be applied with having a very low intensity and short duration, very short duration. And you can manage this duration, intensity, either it is 4 volt, 4 millivolt or 5 millivolt, you can manage it. You can control it by this. And if you require only single stimulus, that provision is there. Whenever it is made, the switch is on, then it will produce a single stimulus. In the output electrode, there will be a single stimulus and for a short duration and according to your choice of intensity. If you want more than one stimulus, that sometime we will be experimenting with repeated stimulus to see what happens to the muscle when it is repeatedly contracted, what happens. So for that purpose, so this is the stimulus. From here we can send the appropriate stimulus to the muscles. So if one is recording device we got, one is stimulating device we got, muscle we got, and let's see the other part. Long arm, short arm, this is the fulcrum, this is attached to this muscle, gastrocnemius muscle, this is the sciatic nerve, this is the stimulator, this is the electrode. So when you send a stimulus, single stimulus to this nerve will be stimulated and the impulse will be passing through this and coming here. There will be a pause. Why this pause? Because it has to pass through a gap. What is this gap? Neuromuscular junction. So here the nerve has to secrete acetylcholine from the nerve endings. And this acetylcholine has to bind with the post-junctional membrane, that is sarcolemma. There it will bind and it will activate sodium channels and action potential will be produced. Now this action potential on the sarcolemma means skeletal muscle membrane. Here an action potential will be produced. Now this action potential will be traveling and then it will enter into the T-tubule. Then from the T-tubule and cistern, there will be release of calcium ion. And now contraction, excitation, contraction coupling will be made. Then only muscle will contract. So firstly we can see, whenever we get the give the stimulation here, so what are the events? Transmission, conduction, passing through the neuromuscular junction and producing contraction, excitation, contraction, coupling, then contraction. So this may take some time, starting from conduction. Okay, another thing is there. This muscle, when it will be contracted, this weight and weight of this has to be lifted. I am lifting my hand. Easily I can do it. Now if I place some weight, then the process will be slightly delayed because of this weight. So this stylus, these are also having some weight. If the weight is more, then it will take more time to produce the contraction. If the weight is less, it will make less time. So what we will be doing before this experiment, we'll remove this. Just you there is a screw, so unscrew it, then you can move it away from the drum. Okay? Now we do one thing, we have to take a time tracing using the tuning fork. Using the tuning fork. How you can do it? This is the tuning fork, isn't it? It looks like this. So in one arm, I made some attachment. I took a piece of X-ray plate.
Yes, sir.
So from this X-ray plate, take a small piece. And this much plate, this much width. Then you, one end, make it sharp and carve it. Then the other end, fix it with one arm of the tuning fork. Okay? Now if I vibrate it, make the tuner vibrating, so there will be vibration of the tuning fork. So you switch on the drum, it will start moving. Then you touch the curved portion onto the surface of the drum. So what will be there? You will get a tracing. What type of tracing? This is called time tracing. How you got it? This is using the tuning fork. How you have used the tuning fork? I explained. Okay. Here one cycle, 100 cycle means 1 second. 1 cycle means 0.01. So from the tip of this cycle to tip of this is one complete cycle. So this distance is this much. You can also mark from here to here. This is same. Okay? Now see this. This is the tracing you can have. When you give the stimulus to the muscle, it will make contraction. This type of contraction you get. And here you will find 3 time periods. Here you will get 3 time periods. What is PS at the first? PS means point. S means stimulation, point of stimulation. So at this point, you gave the stimulus. And other points are easy. From which point contraction has started? PC. And when is the maximum? This is the point of maximum contraction. So you draw a vertical line. Since it is the maximum point of maximum contraction, so this is the time period. This is the time it took to contract. And this is the time it took to relax. So how many? This is one time period. After stimulation, there is no contraction. You put stimulus, but there is no instant contraction after some time. So this time gap is called latent period. What is this period? A latent period. And we'll...
PS to PC is latent period.
PS to PC is the latent period. Why this latency? We'll explain. Why this latency? Sir will explain. So just wait. PS to PC is the latent period. And sir will explain, as I said, why this latency. Then, what is the next period? Period of contraction. It starts from PC, ends in PMC. Okay. How much time it took for contraction? How many cycles are there? 1, 2, 3 means how many seconds? How much time? 103. And what is the time taken for relaxation? 104 seconds. Not only 0.04, 0.04 second is the relaxation. And what is the latent period? 0.01 second. During your examination, we will give you only no pointing. Only one point will be there. One mark will be there. When we gave the stimulus, no other pointing will be there. So you have to tell, seeing this graph during your exam, only the graph you will be having, then there will be some question. What is this twitch? What is this graph? So you will write, it is a simple muscle twitch of gastrocnemius muscle of frog. Okay, more precisely, gastrocnemius muscle is a skeletal muscle. Within bracket, you can write the skeletal muscle. Okay, so it is a skeletal muscle contraction. And how you got it? After stimulating on the nerve supplying it. Okay, and you have to identify the question, what are the different periods? So first you have to point it out, then you have to write there is latent period, there is contraction period, there is relaxation period. Then you will be asked how much is this time. Okay. Now last part, you explain what is happening during latent period, first period, what is happening during the second period, what is the happening third period. So what you have learned from this? If a stimulus is given to a muscle through nerve, what will do? It will be responded by making a twitch. That is the thing. Very first slide. What is our aim? Recording of simple muscle twitch, simple muscle contraction. Okay, of, more precisely, of gastrocnemius muscle of frog. Okay, and what is the overview? A single brief electric shock of adequate stimulus, means of threshold value, applied to the nerve of a skeletal muscle, when you applied such stimulus, it will produce a brief contraction. The muscle will produce a deep contraction followed by relaxation, and the whole thing is called simple muscle twitch. Okay, what are the required things? These are the required things. Then next slide. What are the components? What we will see in the curve? You got this? So you go through. You will get the latent period. What is this latent period? From the point of stimulus to the point of onset of contraction. Unless you mark the point of stimulus, you cannot determine it. Can you determine? So it has to be given to you. Where is the point? Then only you can calculate the other point. So then duration of latent period depends on certain factors. What are these? Conduction of impulse along the nerve, I already told. This is the nerve and here is the muscle. So I have given the stimulation here. So this action potential nerve impulse has to be transmitted along this. So this will require certain period of time. Then second point, the certain time will also be taken for transmission across the neuromuscular junction. And third point is for excitation-contraction coupling. After calcium release of calcium ion, then only initiation process of contraction. This is called very important for your exam, coming exam in the March. EC coupling. Mechanism of EC coupling in skeletal muscle. If you write only mechanism of skeletal muscle contraction, no. Specific answer I need. What do you mean by excitation-contraction coupling? So, what is happening during this excitation contraction? You have to explain it. If I ask for what is the mechanism of contraction of, mechanism of contraction of, that is the whole thing you have to mention. Then third point is, there is what? There is tendons. Tendons are made of elastic tissues. So there will be stretching. When it will be contracted, there will be stretching on the muscle. So this will also take some time. And lastly, inertia, weight of the arm, long arm. Okay, so if the inertia is, the weight is more, the inertia will be more. So this will take more time. So what are the, if I ask what are the causes of latent period? So you have to write all these 5 points. Again you go through it. If you don't understand, please tell me. Okay, then what are the latent period? What is next? Contraction period. You know from where to which part to what is happening during muscle is contracted. Okay, and relaxation period, muscle is relaxed during this period, and maximum height of contraction. So maximum point, peak, how much, some contraction may be, you have applied some drugs, then again you applied a stimulation, what you got? Height is reduced. Then what is the observation? The contraction will be less. More force, less force of contraction will be generated. Okay, so height is important. With the height, you can measure how much force is generated. If his height is more force is generated. If the height is less force is generated. So all these 4 points you have to remember. Okay, I told you, you find the peak. I told you to find the peak, then you draw a vertical line. If you draw a vertical line, this will cross this baseline here, not here. What you will do? Whether you will follow this, you will draw a curve, a line from there this way. It's just like an arc only. Arc. What is arc? Part of a circle. So this is nothing but part of a circle. Actually, how it has been done? You pull it. Hello, look at this. You go on pulling it, and this stylus will be going up. From here, it will not be touching this point. This will come this way. Because this is an arc, this is the center of the fulcrum. So this will move like this. As a result, you will create a curved line. So during the experiment, what is actually done, after it is over, the drum is brought at the midpoint, then here it is pushed or it is just pushed upward, then this arc is made. So with this arc, you cannot find the vertical line. So you have to draw a vertical line if you want to calculate exactly the contraction period and the relief. From the peak, you just draw a vertical line. That will be the actual. Then only you will be able to calculate the actual contraction. Any doubt?